I don't want that, but yeah. Okay, can somebody confirm you can hear me?
I
I
can
can
hear
hear you.
Thank you. Before we begin, are there any questions?
Is the best way to reach you via Canvas email?
Yes, or you can ask in Zoom. Um, there is somebody who wanted a meeting. I need to get to them. Are you the person who wanted the meeting with me?
I know, yeah, I emailed you. I'm not sure if there was others.
Okay. Um, I'm sorry I didn't reply back, but we can meet either after the lab or sometime, well, after the lab or even during the lab if you're okay with students hearing the conversation. Um, that's what I meant to send you, and I just never got around to doing it. Sorry. Uh,
No,
so today is July 31st. We're going to be hopefully finishing chapter 4 and beginning chapter 5. There is a lab today. Any questions about what we're going to do? Am I sharing my screen?
Yes.
Thank you.
I thought we already started chapter 5.
Are we ahead of the schedule? Or did I look at the date wrong? July
today's the 30th. It says chapter 5 on the.
thing. Today's the 31st.
Oh, yeah, the 31st.
So let me think. Is that the wrong date that I have there? Let me look at that.
Well, it says chapter 5 going into 6
Yeah,
during.
it should be. Today's the 31st. That's the wrong day, I think. That should
Well,
be
it says the
Tuesday.
30th through,
The
through.
3rd. Yeah, I
Or,
know, but this should be Tuesday, and it's not Tuesday. The 30th is Wednesday. So that's what the problem is.
oh.
Let me change that now. Let's see if August 6th is correct. Yep, I got all of these dates wrong. Sorry about that. I'll try and correct that later. I've only got the first two corrected there. All right, so today's the 31st, so we're supposed to be working on chapter 5, hopefully finishing it and beginning chapter 6. I wonder how far off I got on that, but I'll look at that later. So let's move on to chapter 5. Oop, I'm on the wrong one. That's what the trouble is. That's the summer 2024, not summer 2025. Yep, that's why I have the wrong syllabus here. Let's close that down. I think it's the same other than the dates. Let's go here. Yep, there it is there. So, yeah, we're supposed to be finishing chapter 5, beginning chapter 6. Uh, screw. Actually, let me close this down. My computer is working slow. Probably because I've had it on for too long. All right, I have that we were talking about the nutritional classification of organisms, and I mentioned you need to know everything in red. The things that are red are useful to know, but you don't need to know it. like the photoautotrophs can be split into, let me blow this slide up. I get it. Can be split into the oxygenic and the non-oxygenic, the anoxygenic photoautotrophs. The oxygenic are phototrophs that release oxygen, and all green plants, algae, cyanobacteria, many green bacteria are oxygenic, but there are some anoxygenic photosynthetic bacteria. For example, the green and purple sulfur bacteria. And then the chemoheterotrophs, you can split them into where the final electron acceptor goes, meaning where the electron ends up from, well, we'll start with glucose in aerobic respiration, and it'll go to oxygen. But anaerobic respiration, the electron from glucose does not go to oxygen. It will go to some other molecule. And we'll talk a little bit about that when we're talking about anaerobic respiration as well as fermentation. Any question about any of that? Go back to the side if I can. Well, that'll be good enough. Nutrient molecules have energy associated with the electrons that form their bonds. And that is how we get our energy when we eat our food. We break the bonds in the molecules of food, releasing that energy, which much of it will be stored as ATP, the energy in ATP. In cells, energy is often obtained from catabolic reactions and stored in the bonds of ATP. And for example, when we eat our food, we're getting our energy from the catabolism of the molecules of food, such as glucose, and we burn that glucose to CO2 and water, and the releasing energy will be stored in the bonds of ATP. ATP is the energy carrier of cells. So when cells normally need energy, they obtain it from ATP. ATP has a high energy unstable chemical bond in the third and the second phosphate group bound to the ATP. And when that chemical bond is broken, it releases a lot of energy, which can be easily used by the cells for its energy needs. So we call ATP the energy carrier or the energy currency of cells. And by currency, we mean like money is the currency we used to buy everything. Obviously, now you got digital coins and all sorts of stuff. But in the old days, money was the currency. And if ever you wanted to buy something, you either had to pay for it by check, which was another form of money, or using cash itself. And so ATP is the energy currency of cells. Any question about any of that? So when cells produce their energy, they do it by oxidation reduction reactions. We'll talk a little bit about that. Oxidation is where one atom or molecule loses an electron, and reduction is where another atom or molecule gains an electron. Oxidation reduction reactions are always coupled. You always have one molecule or electron losing the electron, and then another molecule or electron gaining the electron. Any question about that? Oxidation reduction reactions was a little bit of a big word for, I guess, biochemists, so they shortened it to redox. Redox reaction is the same as oxidation reduction reactions. Any question about any of that?
What are some examples of this?
All right, well, here's a generic example where A is losing an electron. It's gained by B. So A becomes oxidized, losing the electron. B becomes reduced. A more specific example is we have copper +1 and iron +3. Copper loses an electron, so it becomes more positively charged, and it's going to be changed to copper 2+, so copper is oxidized. And then iron gains an electron, and it was 3+, it now becomes 2+, because an electron has a negative charge. And so iron is reduced. Any question about any of that? All right, so an easy way to remember oxidation reduction reactions is the mnemonic Leo the lion goes grr. Leo, lose electrons, oxidation, so L for lose, E for electron, O for oxidation. So Leo the lion goes grr, grr, G for gain, E for electron, R for reduction. Any question about any of that? I didn't move. So in biological system, oxidation is usually more than just the electron. The protons, meaning the H plus ion, is usually removed with the electrons. And then the proton usually follows the electron. So therefore, biological oxidations are often called dehydrogenations because the electron is removed and then follows the electron. Excuse me, the hydrogen is removed from the, we'll say, molecule here, and then follows the electron. So let me blow that up so I can see it a little better. So we have an organic molecule such as glucose, and it's going to lose an electron to NAD+, which is actually the coenzyme in aerobic respiration. The molecule losing the electron will be oxidized, and it will lose an negative charge, but it also loses a hydrogen. So in this case, it's no change in the charge. And then NAD+ actually gains 2 electrons, and it gains a hydrogen plus ion. So it becomes no charge, NADH. And so NAD+ becomes reduced, NADH. And then the extra hydrogen ion is just sort of following the NADH. It's not bound to the NADH. You don't need to know about the hydrogen ion. And then the organic molecule, which I'm just saying, let's make it glucose, which loses 2 electrons, becomes oxidized. Any question about any of that? All right. So biological redox, meaning oxidation reduction reactions, are used in catabolism to extract energy from nutrient molecules. Highly reduced compounds, that's a compound having many hydrogen atoms attached to it, these molecules are degraded, that's catabolized, to a highly oxidized compound. So glucose, C6H12O6, has a lot of hydrogens attached to it, so it is a highly reduced compound, and it loses the electrons. So it's oxidized, and then it's converted into CO2 and water. The energy that's released from glucose can be trapped to make ATP. Not all of it, but much of it. Not even most of it, but what is it, like 40% of the energy released from glucose can be trapped and stored as ATP. So highly reduced compounds contain a lot of potential energy. And when you burn them, they release that energy. For example, if I had a cup full of glucose, hopefully you can see this. I don't see that. Well, now you can see it. Had a cup full of glucose. In order to take a blowtorch to it, I could burn that glucose. I'd have to stand several feet back because of so much energy being released from the glucose. And then from another proof about glucose having a lot of energy in it, people who make homemade bombs often use glucose as one of the ingredients to make the bomb. Because when the glucose becomes oxidized, releases its energy, a lot of energy is released. Any question about any of that? And while the energy is stored in glucose, it's stored as potential energy. The energy in the the chemical bonds of glucose. So let's talk about aerobic respiration, the way our cells get the majority of their energy, and the way many microbes, not all, get a lot of their energy as well. Aerobic respiration is the most efficient way of converting the energy in a food molecule, such as glucose, to the energy in ATP. So with aerobic respiration, we have glucose, and it combines with oxygen. In cellular respiration, which is also called aerobic respiration, and I prefer aerobic respiration because cells can perform respiration, but it could be anaerobic respiration. And it's not called cellular respiration because cellular respiration specifically means aerobic respiration. So I don't use that term, but it's shown here, so you kind of should know cellular respiration means aerobic respiration. Some of the energy in glucose is lost as heat. What we mean is that energy stored in glucose is converted to the energy of heat. And some of that energy can be converted into the energy stored as ATP. And then when the cell needs energy, it can break down the ATP to use the energy in ATP. And then glucose and oxygen are converted into carbon dioxide and water. Any question about that? So the summary reaction for aerobic respiration is C6H12O6, that's glucose, plus 6 molecular oxygen molecules, plus 38 ADP, adenosine diphosphate, and 38 phosphate, the I just means an inorganic phosphate, is converted into 6 carbon dioxide molecules, 6 water molecules, and 38 ATP molecules. This is the summary reaction for aerobic respiration because aerobic respiration takes a lot of chemical and physical reactions to occur. Any question about any of that? So some of the energy from some redox reactions is used by the cell to make ATP. And by I mean some of the redox reactions, remember in humans and all animals, there's only two, aerobic respiration and fermentation. That can be used to make ATP. Animals are pretty lousy at making ATP. And even green plants, which are better at making ATP, meaning they have, I don't know, maybe a handful of different reactions to make ATP. The majority of their catabolic reactions, the redox reactions, cannot be used to make ATP. Any question about any of that? All right. ATP is generated by the phosphorylation of ADP. Phosphorylation is just the addition of an inorganic phosphate to another molecule, such as ADP. So here we have ADP, also known as adenosine diphosphate. That's why the ADP, A for adenosine, D for di, and then P for phosphate, so diphosphate. You need energy to enter this reaction because ATP has more energy than ADP, and then you combine the inorganic phosphate to make ATP. And this energy comes, at least in our cells, from either aerobic respiration or fermentation. And we'll talk about fermentation later. So for now, it just comes from the breaking down of glucose, which it doesn't matter if that's aerobic respiration or fermentation. Any question about any of that? Let me bring this up now. All right, ATP has two high energy bonds. This third phosphate connected to the second phosphate is a high energy bond, and the second phosphate connected to the first phosphate is also a high energy bond. Whenever the cell needs energy, it can break off this phosphate to release this energy in that high energy bond, and then use that high energy bond or the energy in it for its energy needs. If the cell needs further energy, it can break off the second phosphate group. Any question about any of that? All right.
Unstable bonds, you said?
It's a high energy bond.
Okay. I was just thinking back to a couple slides back when you said, I thought you had said they were high energy and unstable.
I didn't really say unstable, but they're not
Okay.
as stable as a low energy bond. I will
Okay.
admit that. But they're not unstable.
Yeah.
Meaning when ATP is made, it stays around. It doesn't self-break down
Okay.
until the cell wants to break it down, in which case it uses an enzyme to break it down.
Perfect.
All
Thank
right.
you.
The generation of ATP is critical for cells because they need it to perform their work, the work the cell wants to do. And by work, I mean the cell does something that requires energy. Cells have three mechanisms for making ATP. There's substrate level phosphorylation, where you can transfer a high energy phosphate from a phosphorylated compound directly to ADP, like a tricarbon phosphate having a high energy bond with that phosphate can transfer that bond to ADP to make ATP. This molecule actually happens in fermentation or this chemical reaction actually happens in fermentation and glycolysis. We're not going to talk about it much as a substrate level phosphorylation, but I will show you the chemical reaction in glycolysis and in fermentation, making ATP. Any question about any of that? So that's the first way cells have for making ATP. The second way cells have for making ATP is respiration, also called oxidative phosphorylation, and I don't use this term. I will just use respiration. Notice I did not say aerobic respiration because there are different forms of respiration. Aerobic respiration is one form of respiration. Respiration involves the sequential transfer of electrons from an electron donor molecule, such as glucose, to a final electron acceptor. And in aerobic respiration, the final electron acceptor is oxygen. Along the way, the electrons give up some of their energy to other molecules in the electron transport chain. And so we call these, I guess, intermediate electron intermediate electron acceptors. And so oxygen gives it to NADH. NADH contains the electron, takes it to the electron transfer chain. We're not really going to talk about FMN, but we'll just call it the first protein in the electron transport chain. And then that electron flows to the next protein in the chain, to the next, 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 and then leaves the electron transfer chain, flowing to oxygen in aerobic respiration. So oxygen is the final electron acceptor. And along the way, the energy in that electron is released in a stepwise fashion. And that's why the cell does not get fried by losing the electron from glucose. Like I said, if I had a cup of glucose, I could get all the energy in glucose to be released by taking a blowtorch to the glucose and causing it to catch on fire. And so much energy would be released as heat, I'd have to stand at least 6 feet back. In a cell, the energy is released in steps, and that way it doesn't fry the cell. Any question about that? And as the energy is released, that energy can be used to generate ATP by chemoosmosis, or we're just going to say aerobic respiration. And it doesn't matter if it's anaerobic respiration or respiration, the electron carriers carry the electron from glucose to the electron transport chain. It could be an aerobic respiration electron transport chain, which is actually shown here, where the electron will end up at oxygen. But it could be another electron transport chain, which will generally be shorter. And then the electron will not transfer to oxygen. It will transfer to some other molecule. Any questions about any of that? All right, so that's the second way that cells can make ATP, by respiration. The third way that cells can make ATP is by photophosphorylation. This only occurs in photosynthetic cells. Light energy releases the electrons from chlorophyll, or another photosynthetic pigment, and then that electron moves, and that light energy, by this electron moving through an electron transport chain, can be converted to chemical energy, making ATP and NADPH. However, the ATP and the NADPH are often used by the cell to make sugar. So most people, when they're talking about photosynthesis, they say it happens in the presence of light, so the energy comes from light, and then you take in CO2 and water, and then you make sugar. Well, along the way, you do make ATP and NADPH, but these molecules are broken down to make the sugar. Any question about any of that? And in this picture, you can see photosynthetic organisms. That's why the boys are so muddy. Only kidding, the photosynthetic organisms are in the background. All right, any questions about the 3 ways that cells can make ATP? So the energy is released from molecules, like molecules of food, in controlled ways. and then stored by a series of reactions so that the energy is not released in a single burst. Because if it was released in a single burst, your cell would be fried. It would be actually charcoal because it's so hot. Electrons from the donor molecules, such as glucose, are passed from one compound to another through a series of redox reactions. Let me show you that there. Which I already showed you, but let's go through it here. So the first, glucose is the first molecule, and it's going to lose the electron, so it'll become oxidized to NAD+, which will become reduced and become NADH, and then it'll give its electron to FMN, which will be oxidized, loss of electron, and then FMN will gain the electron, so it'll be reduced. FMN will then give the electron to the next protein in the electron transport chain. So it will lose the electron, so it'll become oxidized, and then the next molecule become reduced, gaining the electron, etc. Any question about any of that? All right. Carbohydrate catabolism is a way that many organisms use for getting the energy they need or for making ATP. So most organisms can use carbohydrate catabolism as their primary source for cellular energy. The most common energy source is glucose, but they may use lipids, proteins, or any other food molecule. I guess carbohydrates would be another one. We'll talk a little bit more about that later. To produce the energy from glucose, microorganisms use two general processes to make ATP. They can use either respiration or they can use fermentation. In respiration, you should know there's two types of respiration: aerobic respiration, which uses oxygen, meaning the electrons flow to oxygen. And then there's another form of respiration, anaerobic respiration. And there's actually many different types of anaerobic respiration where the electron flows from glucose to a molecule other than oxygen. And it just depends on what form of anaerobic respiration it is, what that molecule will be, where the electron ends up. It could be nitrite, could be nitrate, it could be sulfate, it could be hydrogen sulfide gas, it could be iron, an iron compound. Any questions about any of this? If not, let's watch a little video on aerobic respiration. Might take a while for this to load. Yep, we got a commercial.
Hey, Moo. iDrive. You help people customize and save with Liberty Mutual.
All cells need energy to stay alive. Usually your cells get their energy.
Let me up the volume.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with the molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into 2 molecules of pyruvate, energizes 2 electron carrier molecules, and produces 2 molecules of ATP. The next 2 stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create 2 more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
Okay, any questions about that video? So here we're looking at an overview of aerobic respiration on the left and fermentation on the right. Aerobic respiration tends to start with glucose, and it goes through glycolysis. Let's say it goes through 3 principal stages: glycolysis, the Krebs cycle, and the electron transport chain. In glycolysis, glucose is converted into pyruvic acid. Glycolysis is followed by the Krebs cycle shown here. And pyruvic acid is actually converted into CO2 in the Krebs cycle. And along the way, the electrons are carried along from glucose are carried by NADH and FADH2. FADH2 is not shown in this picture. And then the electron carriers, NADH and FADH2, carry the electrons to the electron transport chain, the 3rd step of aerobic respiration. And essentially, electrons flow from glucose, which is a high energy rich molecule, to CO2 and water, which are energy poor molecules. And then that energy in glucose is converted to make ATP. In aerobic respiration, we have a high yield of ATP, and that compares with fermentation, which only has glycolysis, and then 1 or 2 fermentation steps after glycolysis, meaning there's no Krebs cycle, there's no electron transport chain. Pyruvate is, instead of converted to CO2, pyruvic acid or pyruvate is converted into the end product of fermentation, like alcoholic fermentation. The pyruvic acid is converted to ethanol. And then the ATP yield of fermentation is much lower than the ATP yield of aerobic respiration. Any questions about this overview? If not, let's go into aerobic respiration. This is showing you aerobic respiration in a eukaryote cell. And the video showed you this in similar terms. They didn't mention this is aerobic respiration in a eukaryote cell because it involves the mitochondria, and only eukaryotes have a mitochondria. So initially in aerobic respiration, we have glucose, which undergoes glycolysis to pyruvate, and glycolysis is happening in the cytoplasm. So if we put this purplish square, the cell, glycolysis is happening in the cytoplasm of the cell. The pyruvate then moves into the mitochondria. And inside the matrix, or inside the center of the mitochondria, that's the matrix of the mitochondria, meaning inside the inner mitochondrial membrane, we have the Krebs cycle, also called the citric acid cycle, which follows glycolysis. And that will convert the pyruvate to CO2. And that happens inside the mitochondria, inside the inner membrane of the mitochondria, also called the matrix. Along the way, NADH and FADH2 are generated both by glycolysis and the Krebs cycle, and they carry the electrons to the electron transport chain, which here is called oxidative phosphorylation. I don't use that term. I use the term electron transport chain, which can also be called electron transfer chain. And students don't tend to get confused on electron transfer chain, electron transport chain, because those are very similar. But oxidative phosphorylation is very different, so that's why I don't use it. And the electrons are brought to the third step of aerobic respiration. And then in the third step, ATP is made at high numbers. But along the way, we have ATP made some, a little bit in glycolysis, some in the Krebs cycle, and the majority of it made in the electron transport chain. The electron transport chain occurs in and along or through the inner mitochondrial membrane of a eukaryote cell. Any question about any of that? All right, for a prokaryote, we don't have a mitochondria. And in a prokaryote, we have glycolysis happening in the cytoplasm, so that's similar. The pyruvate then moves to a membrane, having the enzymes necessary for converting aerobic respiration. And where that membrane is it's the cell membrane, because that's the only membrane that bacteria and prokaryote cells have. The citric acid cycle happens in the cytoplasm near the membrane, cell membrane. And then the electron transport chain happens through and along or across the cell membrane of a bacteria or archaea. Any question about any of that? All right. Glycolysis is also called the Epstein-Meyerhof pathway, after the two German scientists who discovered it. I may be butchering these names because I'm not German. I always call it glycolysis. In glycolysis, we have the oxidation of glucose, which is converted into pyruvic acid. Glycolysis occurs in most living cells. Glycolysis can occur in the presence or in the absence of oxygen. Glycolysis does not use oxygen, but it can occur if oxygen is present. Any question about any of that? Glycolysis does produce ATP and NADH, but not very much, just a little bit. Here is the, the chemical reactions or the chemical pathway for converting glucose, a 6-carbon molecule, in glycolysis to pyruvic acid, a 3-carbon molecule. When I was in undergraduate school, I needed to, as an undergraduate, needed to memorize glycolysis. And not only did I need to know every intermediate molecule, but I needed to know the names of the enzymes, which were converting that molecule to this molecule. And I think it was fair that since I had to learn all that stuff, that I have to require you guys to know all this stuff. Although not necessarily the enzyme name, because it's not even presented here. What do you guys think of that?
So do you want us to, like, what you said, not the enzymes, but like every like equation?
I'm kidding you, okay? You
Okay.
don't need to know any of the intermediate molecules. You do not need to know the chemical equation of glycolysis. You should know the summary reaction shown here, that you start with glucose. Actually, you should know what's in red. You get a net gain from glycolysis of 2 ATP. By net gain, I mean there's a net gain because glycolysis uses 4 ADP and makes 4 ATP, but you require 2 ATP to get glycolysis to run. So we get a net gain of 2 ATP from glycolysis. The glucose is converted into 2 pyruvates, also called pyruvic acid. The difference, you need a chemist to tell them apart. Pyruvic acid has the hydrogen attached to it, but pyruvate does not. The cells can use this molecule and that molecule the same, so I tend to treat them synonymously, and a pure chemist wouldn't like that. But for our purposes, we can treat them synonymously. And then 2 NADH are made by glycolysis. So you do need to know this. It wouldn't hurt to know this equation, but you do not need to remember this equation. And that is in glycolysis, you take glucose, you add in 2 ATP. Let me show you where that is. You add in the first one there, and then the first one there. But then you get out 4 ATP, 2 here and 2 there. So there's a net gain of 2 ATP made by glycolysis. Any question about any of that? And then you take in 2 NAD plus molecules to generate 2 pyruvic acid molecules, 4 ATP or a net of 2, and then 2 NADH molecules. You also have 2 hydrogen ions and 2 water molecules. You don't really need to know, well, you don't need to know any of this, especially these 2. Okay? Now, when we're talking about respiration, you should remember it's the oxidation of molecules, releasing electrons, which then are taken to the electron transport chain to generate ATP. But when I use the word respiration, remember that I mean both aerobic respiration, where oxygen is the final electron acceptor, and anaerobic respiration, where the final electron acceptor is not oxygen, it's some other molecule other than oxygen. And there are different forms of anaerobic respiration. It just depends on what the final electron acceptor is. Along the way, in respiration, ATP is generated. So let's talk a little bit more about the preparatory step in the Krebs cycle. From glycolysis, we have pyruvic acid, but pyruvic acid cannot directly enter the Krebs cycle. The pyruvic acid is therefore oxidized and decarboxylated. Coenzyme A attaches to the oxidized and decarboxylated pyruvic acid, meaning it's a molecule smaller than pyruvic acid. CO2 is given off because pyruvic acid is a 3-carbon molecule, and the end of the preparatory step is acetyl coenzyme A. It's a 2-carbon molecule. And then coenzyme A is attached to the remains of pyruvic acid. Oxygen has not been used yet, but we are generating CO2. And this is the first CO2 made. Now, the preparatory step runs 2 times for every molecule of glucose because in glycolysis, we convert, where do I have that? We convert glucose to 2 pyruvates. So 1 glucose molecule becomes 2 pyruvate molecules. The pyruvate are 3 carbon molecules, the glucose 6 carbon molecules. Where was I? So therefore, the preparatory step always runs twice per molecule of glucose. Any question about that? All right, let's go on to the Krebs cycle. The Krebs cycle has different names. It's also called the tricarboxylic acid cycle because it has a lot of 3 carbon units, which usually are together to form 6. And it's also called the citric acid cycle because the first molecule made in the Krebs cycle is citric acid. And these are abbreviated TCA for this one, T, tri, C, carboxylic, A for acid. And then this one's abbreviated TCA also, the T for the, C for citric, A for acid. Any question about any of that? Now, you kind of need to know this just for the book when you're reading through. I will always use it, the Krebs cycle. And your textbook does tend to use the term Krebs cycle. But there are other terms which mean the same as the Krebs cycle of aerobic respiration. Any question about any of that? And these are the, well, 2 counting the abbreviation. All right, here we have the preparatory step shown right here, this one step, changing pyruvic acid into acetyl coenzyme A. The acetyl coenzyme A combines with oxaloacetic acid in the Krebs cycle to make the citric acid. You don't need to know this, but it's the first molecule made in the Krebs cycle. And then the citric acid is converted to this molecule. That molecule is converted into this molecule. NADH and CO2 are generated in this step. And then this molecule is converted into that molecule. Once again, NADH and CO2 are generated in this step. And then we have this step of the Krebs cycle, which actually generates the ATP of the Krebs cycle. And then we have the next step, which generates some FADH2. And then the last step, you don't need to know this, but converting malic acid to oxaloacetic acid. And in the process, NADH is generated. So NADH is generated in the preparatory step in the Krebs cycle 5 times, 1 here, 1 there, 1 there, 1 there, and that's actually not NADH, that's FADH2. And then 1 here, NADH. So 4 times for the NADH, and then 1 time for FADH2. The overview of the Krebs cycle is you start with pyruvic acid. It is then used to make ATP, and you get 1 ATP for each molecule of pyruvic acid. But remember, for glucose, both the preparatory step and the Krebs cycle will run at least 2 times. Any question about any of that? All right. And then 4 NADH and H plus are made. There's the first NADH, there's the second, there's the third, and there's the fourth. 1 FADH2 molecule is made right here. And in the process, CO2 is generated. All of the carbon in pyruvic acid is changed into CO2. Let me blow this up so I can see it a little better. There's the first CO2 being generated in the preparatory step. There's the second CO2 generated in the Krebs cycle. And there's the third CO2 generated in the Krebs cycle. Remember, you have to have 1 carbon for each carbon in pyruvic acid. And so there are 3 carbons here, and they're converted into 3 molecules of CO2. Any question about any of that? And that's why we breathe out CO2, because our cells are engaging in aerobic respiration, burning glucose and other foods into CO2 and water. Any question about any of that? That's why water stays on the planet. It's not used up by aerobic respiration, and it will be generated pretty much from where it started, but that might be several generations ago. All right, NADH and FADH2 are made by the Krebs cycle or whatever. And it's 4 NADH and 1 FADH2 per molecule of pyruvic acid, or acetyl coenzyme A. However, if I were to ask the question per molecule of glucose, you need to multiply each of these by 2. And how you can see that is 2 times 3 is going to be 6. And glucose has 6 carbons in it. And every one of those carbons is converted into CO2. None of it is converted into oxygen. Any question about any of that? All right, the last thing I need to talk about is the Krebs cycle is called the cycle because we're ending up at the molecule we started with. And then the cycle just repeats and repeats until the day you die. Any question about any of that? All right, NADH and FADH2 take the electrons, or they're said to carry the electrons to the electron transport chain. There's the overview of aerobic respiration here. We'll talk about fermentation later. So the electron transport chain, abbreviated ETC, is a series of carrier molecules that are in turn reduced and then oxidized as electrons flow from glucose and are passed down the chain. Energy is released as the electrons are transferred from high energy to lower energy compounds. Note, I'm saying high energy, but I'm not saying low energy, because this energy here tends to be very similar to the molecule carrying the energy released by glucose. And then energy is released by chemiosmosis to produce ATP. I don't often talk about chemiosmosis, but that's the step of the preparatory step that is used to make pesto. All right, any question about any of that? Let me see if this link will work. Seems to be working.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Okay, any questions about that video? So this is showing you the electron transport chain. Remember, in eukaryotes, this occurs along the inner membrane of the mitochondria. In prokaryotes, the electron transport chain occurs along the cell membrane. In the electron transport chain, the electrons are carried from glucose by NADH and FADH2. NADH is more efficient because it brings the electrons to the first protein in the chain of proteins in the membrane. FADH2 is not as efficient because it brings it to the second protein in the chain of proteins in the membrane. The first protein takes the electrons and then moves the electrons through the protein. That gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. The electron then flows to the next member of the chain of proteins in the membrane, and then flows to the next protein, shown here. With this protein, the protein uses the electrons moving through it to pump hydrogen from this side of the membrane to that side of the membrane. And then the electrons flow to this protein in the membrane, which then flow from this protein to the last protein in the membrane. And in this protein, we also pump hydrogen ions from this side of the membrane to that side of the membrane. NADH is more efficient because it has 3 proteins that can pump hydrogen from this side of the membrane to that side of the membrane. FADH2 only has 2 proteins that can pump hydrogen from this side of the membrane to that side of the membrane. The electrons then leave this last protein in the chain of proteins in the membrane, and the electrons in aerobic respiration flow to oxygen. So oxygen is the final electron acceptor, taking 2 electrons and 2 hydrogen ions, and that forms water. And this is why we have to breathe in oxygen. All of the oxygen you breathe in will be converted into water by aerobic respiration in the electron transport chain of aerobic respiration. None of that oxygen you breathe in is converted into CO2. All of the CO2 that you breathe out, I've already shown you. I think I've already told you as well. All the CO2 that you breathe out comes from the CO2 produced in this step of the preparatory step in the Krebs cycle. Let me blow this up. So all the CO2 you breathe out comes from the burning of glucose, which will make 2 pyruvic acid. That's 3 carbon molecules. There's 1 carbon dioxide. There's the second carbon dioxide. There's the third carbon dioxide, meaning all the carbons of pyruvic acid are converted into CO2. And then glucose is converted into two pyruvic acids. So all of the carbons in glucose will be converted into CO2. Any question about any of that? Let me go back to where I was. Is it there? No. Kind of here. In aerobic respiration, remember, oxygen is the final electron acceptor, the last molecule to accept the electrons. Along the way, hydrogen ions are pumped from this side of the membrane to this side of the membrane. That means there will be more hydrogen on this side of the membrane than that side of the membrane, and there will be more positive charges on this side of the membrane than that side of the membrane. That creates a twofold gradient. Hydrogen ions will want to move from this side of the membrane to that side of the membrane just by the laws of second law of thermodynamics. However, the hydrogen ions can only move across the membrane at one point, this protein here. And when the hydrogen ions move through this protein, they give this protein the energy it needs to phosphorylate ADP, meaning attach phosphate to it, to make ATP. Any questions about any of that? The conversion of the energy of glucose into the energy of ATP is 40% efficient, meaning 40% of the energy in glucose will be converted to the energy of ATP. You might be thinking that's not very efficient, but guess how efficient the engine in your car is for converting gasoline energy, potential energy in gasoline, into the energy of motion of your car? Anyone have a guess? Relative to 40%, the efficiency of your cell in converting glucose into the energy in ATP? Nobody has a guess? Your car engine is only about 20% efficient. The very best engines humans have ever made are only 25% efficient. And that would be some like plastic engine running a plastic car, which you could never drive because it wouldn't be safe. And the engine would obviously break down over time because it's made out of plastic.
90%?
No, no, no. I just told you the answer. Your car engine is only 20% efficient in converting the energy in gasoline to the energy of your car's moving.
Okay, I misunderstood. I get it now.
And so your cell is actually very efficient compared to the best motors humans can make. Because the best motor we have is about 25% efficient. And like I said, this is a motor you could not use on your car because it would eventually break down. I don't know how long, but it's only used to make like a plastic experimental car. All right, let's go on and see about this. So ATP molecules are produced by the electron transport chain in aerobic respiration. In eukaryotes, we get 32 to 34 ATP molecules made by the electron transport chain. In prokaryotes, bacteria and archaea, we always get 34 ATP made, and this is per molecule of glucose. Most of the cells in your body can only generate 32 molecules of ATP per molecule of glucose, but your heart, the liver, and the kidneys can generate 34 molecules of ATP. What do you know about the heart, the liver, and your kidney compared to other cells in your body, like your skin cells or your nose cell or even your muscle cells, other than your heart muscle? What do you know about your heart? What do you know about your cells in your brain, which can only generate 32 molecules of ATP? Why do you think your cells in your heart make 34 and they're more efficient than the cells in your brain?
Because it's essential to life.
Well, that's true. It is essential to life, but
Okay,
no,
like
the heart is always working. Your brain is not always working, which should be obvious to you now. Being a little bit facetious here, but you're getting the point, hopefully.
Yep.
That means your skin cells are not going to need to always work, and so they're not as efficient. But your heart has to always work. Once it stops working, guess what? You're no longer alive. And that's true of your kidneys and your liver. They're always working. The kidneys are always filtering the blood, removing toxins and waste material from it. And the liver is always filtering the blood, removing toxins and degrading them and other things like that. So the heart, the kidneys, and the liver are always working, and so they're more efficient. They produce 34 molecules of ATP. All the other cells in your body can only produce 32 molecules of ATP per molecule of glucose. Prokaryotes are a little more efficient, and they always generate 34. Remember, the final electron acceptor in aerobic respiration is a molecule of oxygen, and it combines with hydrogen and the electrons to form water. For humans, much of that water will be breathed out, but we can recycle some of that water and it'll be put to use in your body. For some desert animals, they're very efficient at retaining water, and 60% of the water the desert animal gets is actually from aerobic respiration. So 60% of the water these desert animals get comes from simply aerobic respiration, digesting and then burning their glucose to make ATP. Any question about any of that? Let's go on. Well, let me go back one. So here we're looking at respiration. We talked about aerobic respiration, comparing it with fermentation. This is the Krebs cycle, where the electron carrier brings the electrons from glucose to the electron transport chain, and then the electrons move from one molecule in the chain to the next, and then it leaves the last protein in the chain of proteins in the membrane, and the electron flows to oxygen. Oxygen is the last electron acceptor in aerobic respiration, and that oxygen will be converted into water. This is showing you that in eukaryote cells, aerobic respiration starts with glycolysis in the cytoplasm, but then the electron transport chain happens on and in and across the inner mitochondrial membrane. The Krebs cycle actually happens in the matrix inside the center of the mitochondria, but the electron transport chain happens right here. And there you see the electron transport chain, and it's called the electron transport chain because electrons transport from one molecule in the membrane to the next. And it's sometimes called the electron transfer chain just because the electron moves from one molecule, transfers to the next. The electron transport chain is a series of proteins in the membrane, which is the inner mitochondrial membrane for a eukaryote cell. But for a prokaryote cell, it's actually the cell membrane. And as the electrons move through some of the proteins, this one, that one, and this one, the electrons give that protein the energy they need to pump hydrogen ions from this side of the membrane to that side of the membrane. Here's a question for you. If this is the cell membrane of a bacteria, where are the bacteria pumping the hydrogen ions? Is it outside the cell? What's outside the cell membrane of a bacteria? For most bacteria. It's right here. What's that? The hydrogen ions are pumped across the cell membrane into the cell wall. And then the hydrogen ions can move through the cell membrane through this protein here, which is a ATP synthase molecule. It can make ATP from ADP and phosphate. And what it does is it uses the energy of the molecule moving from this side of the membrane to that side of the membrane. Any questions about any of that? Can't get that out of the way. The electron transport chain. Electrons are passed from one carrier molecule to the next in a series of redox reactions. As the electrons pass through the chain, some of the proteins pump hydrogen ions across the membrane. That creates the concentration gradient of hydrogen ions, the twofold gradient, and that's potential energy. The potential energy is used to make the ATP. It's called the proton motive force. I won't quiz you on that. But it's the both the electrical gradient and the increased hydrogen on this side of the membrane, which is the potential energy to make ATP. I don't use this term, but chemiosmosis is the movement of hydrogen ion to make ATP. I just use electron transport chain. The electron transport chain is sometimes called oxidative phosphorylation, and that's because ADP becomes phosphorylated to make ATP. With the complete oxidation of glucose via aerobic respiration, for eukaryotes, we generate 36 to 38 ATP. For prokaryotes, we generate 38 ATP. And there's the overall reaction for aerobic respiration, where we write the step in one step. Remember, it's many different chemical steps. Let's go and show you one chemical step. Here, each of these is a chemical step in aerobic respiration, where one chemical is converted to another chemical. And it also involves physical steps. That is where one state of matter is converted to another state. So a physical change. An example of a physical change is when you put sugar, cream, and milk in your ice cream machine, and then you turn on the machine, and the sugar, the cream, and the milk will be physically changed into ice cream, which doesn't look like or taste like cream, milk, and sugar because of the physical change making the ice cream. And the physical change is actually the movement of hydrogen ion across the membrane. You have both this side and then this side. So there are physical steps and chemical steps involved in aerobic respiration. And the overall chemical equation is C6H12O6 glucose plus 6 oxygen molecules and 38 ADP and 38 inorganic phosphate are converted into 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. Any questions about aerobic respiration? All right, let's talk a little bit about anaerobic respiration. Anaerobic respiration is a form of respiration where the final electron acceptor is not oxygen. It's some other molecule other than oxygen. It could be nitrate, could be nitrite, could be sulfate, it could be an iron compound. It just depends on what form of anaerobic respiration it is. In anaerobic respiration, you do have glycolysis and the Krebs cycle, but you'd also have the electron transport chain, but the electron transport chain is shorter in anaerobic respiration than for aerobic respiration. The ATP yield is also less. Remember, for bacteria and archaea, you generate 38 molecules of ATP. For anaerobic respiration, it's less than that, and the best forms of anaerobic respiration generate 36 molecules of ATP, and the worst form of anaerobic respiration generates around 4 molecules of ATP. So ATP is between 2 and 36 molecules of ATP. It's usually higher than 2, it's usually 4, meaning the worst. Therefore, anaerobes tend to grow slower than aerobes because they do not generate as much ATP per molecule of glucose as an aerobe. Any question about any of that? And the reason why they generate less ATP is the electron transport chain is a little shorter in anaerobic respiration than in aerobic respiration. So here's the electron transport chain. Usually they have less on the start. So the first, meaning the most efficient anaerobic respiration, will be missing this protein. And the worst will only have this protein. It just depends on the form of aerobic respiration. Not aerobic, anaerobic respiration. All right, let's go back to where we were. So comparing respiration with fermentation, respiration always has glycolysis, always has the preparatory step in the Krebs cycle, always has the electron transport chain. Fermentation always has glycolysis, and then it has the fermentation step afterwards. Fermentation only generates a net gain of 2 ATP per molecule of glucose. And then respiration, depending on which form it is something higher than 2 ATP for the worst forms of anaerobic respiration, all the way up to 38 molecules of ATP for aerobic respiration. Fermentation, we do generate ATP, but not much. We do release energy from the oxidation of an organic molecule, such as glucose, via glycolysis. Remember, glycolysis converts the glucose to 2 pyruvic acid molecules. We get a net gain of 2 ATP. We do get some NADH. Glycolysis can also use other molecules other than glucose, but if they're carbohydrates, they'll be converted into glucose and then sent down aerobic respiration the normal way. You can use other molecules as well, and we'll talk a little bit about that when I'm talking about using other food molecules. So I'm not going to talk about that now. So fermentation always involves glycolysis, where we convert glucose into 2 pyruvic acid molecule. We also generate 2 ATP, a net gain of 2 ATP. And then we generate 2 NADH. We then convert the pyruvic acid into the end product of fermentation. For alcoholic fermentation, that would be alcohol or ethanol. And for lactic acid fermentation, we convert pyruvic acid to lactic acid. And that will use up the 2 NADH that were generated here. Now here's a question for you. Like when our muscle cells are engaging in fermentation because we don't have enough oxygen around for the muscle to engage in aerobic respiration. Now, the heart and the lungs are working like mad to generate the oxygen as well as send the glucose to the muscle, but they cannot keep up with the muscles using. So we're talking about like an Olympic athlete, and they will be using their muscles so strenuously or so much that all of the oxygen will be used, and they still have some glucose around. And so their muscles will fall back on fermentation. And I'm sure you've had that happen when you're using your muscles so much that your muscles start burning. That muscle burn is actually from the accumulation of lactic acid happening in your muscle. And what we're doing is we're making ATP by fermentation so the muscle can continue to work because there's no oxygen in your muscle. And so the muscle falls back on lactic acid fermentation to make ATP. And as long as your muscle can make ATP, the muscle can continue to work. If ever the muscle either runs out of glucose or, let me see, is there anything else that would happen? It must run out of glucose because it's not going to run out of anything else. We see this generally about once every Olympics that an athlete pushes themselves to the limit and they can no longer engage in fermentation in their muscles. And so no fermentation, no ATP. What does the athlete do? They just collapse. Without any ATP, the muscles cannot work, and so the Olympic athlete just collapses. The coach will run out in the wintertime and throw a blanket over them. In the summertime, I think they run out and squirt them down with water. And then the Olympic athlete will get up because the heart and the lungs are working like mad, and the muscle cells are given both glucose and oxygen, and so they can engage in aerobic respiration, and then they make ATP, and they get up and then walk across the finish line. I'm sure you've all seen that in an Olympic athlete where the athlete just collapses. No harm to them, as long as the coach runs out and throws the blanket over him in the wintertime or squirts them down with water in the summertime. But that's why our muscle cells use fermentation to generate ATP when there isn't enough oxygen around to engage in aerobic respiration. But the heart and the lungs will be working like mad to try and bring oxygen to the muscle. And my question for you is, why doesn't the muscle cell just retain this NADH and then wait until the heart and the lungs brings oxygen to the muscle cell, and then the muscle cell can use this NADH and generate a lot more ATP than it would if the muscle were to engage in fermentation? Anyone have a guess here? It's actually shown on this slide.
If it continues and uses it, doesn't it just like reproduce it in fermentation?
Yes. If you don't use the NADH in the fermentation step, you do not generate NAD+. And if there's no NAD+, the muscle cell cannot engage in glycolysis and fermentation to generate ATP. So you need to use up this NADH to recycle it so the cell can continue to have NAD plus available to engage in fermentation. Any question about any of that? So you need to use up this NADH to recycle the NAD plus so the cell can engage in fermentation to make ATP. Otherwise, the cell would not be able to engage in fermentation, and the person would just collapse. All right, I've already mentioned fermentation does not use the Krebs cycle, does not use electron transport chain. They do have a fermentation step or two after glycolysis to convert pyruvic acid to the fermentation end product. And there are two fermentation end products we're going to talk about because there's actually a handful of fermentation, what do you call that, processes or methods. We're going to talk about two, lactic acid fermentation. Our muscle cells can engage in it. Many bacteria can also engage in lactic acid fermentation, where you simply convert in one step pyruvic acid to lactic acid. This happens in our muscle cells. It also happens in sauerkraut, in yogurt, and many of our fermented foods. Most cheese, like cheddar cheese, engages in lactic acid fermentation. We actually use the lactic acid to help prevent the food from spoilage. And that's why sauerkraut and yogurt can survive or be good at room temperature for at least a day, if not longer. I'm not sure how long it would be, but at least a day. Yogurt can be at room temperature for at least a day because of the lactic acid in it. And then sauerkraut, the Germans, as well as the Poles and everybody else in Europe who made sauerkraut, they would make it and then keep it like in the Middle Ages, and the lactic acid would preserve the sauerkraut, and then they would eat it all winter long. And that actually was an important source of vitamin C for the Europeans in Europe in the wintertime, because they didn't get fresh fruit in the wintertime. Any question about lactic acid fermentation? Alcoholic fermentation is similar, although it takes two steps. You first convert the pyruvic acid into this molecule, and then you convert that molecule into ethanol. Ethanol is a 2-carbon molecule. Pyruvic acid is a 3-carbon molecule. So 1 carbon is given off as CO2. That CO2 actually gives beer its fizz. In wine, we do make CO2, but it's made in an open casket or an open barrel, and the CO2 is vented to the air. However, if you were to make the wine in a bottle and then cap the bottle, you would have carbonated wine, and we call that champagne. Any question about any of that? Here are the different fermentation reactions. Lactic acid fermentation, which our muscle cells do, many bacteria do. Alcoholic fermentation, which Saccharomyces, a yeast, do for making leavened bread as well as beer and wine. Propionic acid fermentation is the one that is used to make Swiss cheese, and CO2 is given off. That CO2 is what makes the holes in Swiss cheese. And there's a few other fermentation reactions. We can use these to make some industrial products, like butanol can be made by butyric acid fermentation from Clostridium. We can make, acetic acid and lactic acid from Escherichia and Salmonella. And Enterobacter, we can make acetone and formic acid, as well as some other acids and ethanol. Any question about any of this? The only two you really need to know are lactic acid and ethanol, ethanolic or alcoholic fermentation. I won't quiz you on these other forms, but there are a handful of fermentation reactions, and they do use different species, which engage in that fermentation process, and then they make different end products. Here we're seeing some industrial uses for different types of fermentation. Ethanolic fermentation used in beer and wine, also used in making leavened bread. Acetic acid fermentation used in making vinegar. Lactic acid fermentation. This is the one that most of our fermented foods use. Most cheeses like cheddar cheese, yogurt, rye bread, sauerkraut, summer sausage, even chocolate uses lactic acid fermentation. Swiss cheese,
Would sourdough bread be lactic acid?
yes, although sourdough bread uses lactic acid and alcoholic fermentation, meaning you do generate alcohol in sourdough bread as well. So it's using both lactic acid and ethanolic fermentation.
Okay, thank you.
Swiss cheese using propionic acid. You don't need to know that one, but I'm just telling you. We do use some of these others to make different industrial uses, like pharmaceuticals, using molasses, and this one. Glycerol for making glycerol, also using molasses and using Saccharomyces, the yeast. I'm not sure what flavoring this is, but it uses molasses. And then acetic acid, making methane used as fuel. Vitamin C. I don't know of anyone who uses fermentation to make vitamin C, but you can. And that's sorbase fermentation reaction. All right, a summary of aerobic respiration, anaerobic respiration, and fermentation is shown here. The growth conditions, aerobic respiration requires oxygen, so they're aerobes. Anaerobic respiration requires the absence of oxygen, so they're anaerobes. The final electron acceptor for aerobic respiration is oxygen. The final electron acceptor for anaerobic respiration is a molecule other than oxygen. It could be many different molecules depending on the form of anaerobic respiration, like nitrate, sulfate. Some molecule like iron, not oxygen. You don't need to know about the type of phosphorylation. And then the molecules made for aerobic respiration is the most efficient for bacteria, for your heart, your muscle, no, your heart, your kidney, and your liver. You can generate 38 molecules of ATP. For bacteria, it's 38 molecules of ATP per molecule of glucose. For all other cells in our body, it's 36 molecules of ATP per glucose molecule. For anaerobic respiration, it's variable, but it's fewer than 38, but more than 2 molecules of ATP. And it just depends on what form of anaerobic respiration it is, how efficient it is. The most efficient is 36. The least efficient about 4. And then fermentation, we only generate 2 molecules of ATP per molecule of glucose for fermentation. Any question about this? All right. Carbohydrates can be used in aerobic respiration. And for carbohydrates, we generally convert that carbohydrate to glucose, like starch. If you're eating a bunch of starch, you just digest it, and then it's converted into glucose. For something like lactose, the lactose sugar would be broken down into glucose and beta-galactose. The glucose will go down normally. The beta-galactose has to be converted into glucose, and then it will be sent down aerobic respiration as glucose. So all forms of carbohydrate will be converted into glucose and then can be catabolized in aerobic respiration, meaning burn. Most of them will be converted into glucose. A problem with large molecules like large lipids, large proteins, even large carbohydrates is the large molecule cannot cross the cell membrane. How cells deal with these large molecules, including your gut cell and a bacteria, is they extracellularly excrete lipases and proteases outside the cell. And this is true of our gut cell as well. And then you break down the large molecule into smaller molecule, like the proteases will digest the protein into amino acid, and then you bring the smaller molecules across the cell membrane.
Is it time?
Is it time to end? I don't know. Oh yeah, it's time to end. All right, let me go back and we'll continue with this. I was hoping to finish this, but I've got, oh gosh, at least 10 slides to do. All right, I'll see you at 6:30 in the lab. Are there any questions?
No.
All right. All right, I'll see you at 6:30 or whatever time for the lab, 7 o'clock, I think. Yeah.